The sodium-potassium pump, life's molecular engine. Welcome to this explanation of the sodium-potassium pump, one of the most vital molecular machines in animal cells. This protein, also known as Na K ATPase, sits in the plasma membrane and works continuously to keep sodium and potassium ions in the right balance. Every heartbeat, every nerve impulse, every thought depends on the unequal distribution of these two ions. Inside a typical cell, potassium concentration is high, while sodium concentration is low. Outside the cell, the situation is reversed. Maintaining this difference is essential for generating the electrical potential across the membrane. Now look at the left panel of the image. Here you can see a simplified schematic diagram of how the pump works. Each cycle of the enzyme uses one molecule of ATP, the universal energy currency of the cell. As ATP is hydrolyzed to ADP, an inorganic phosphate, energy is released and used to move ions against their concentration gradients. 3 sodium ions are exported from the cell for every 2 potassium ions brought in. This creates and preserves the electrochemical gradient that drives nerve impulses and muscle contraction. Because more positive charge leaves than enters, the pump contributes to the cell's overall negative interior. The reaction sequence begins when sodium ions bind to the inside of the pump. ATP donates its terminal phosphate to an aspartate residue on the protein, specifically aspartate 369, shown in the right panel. This phosphorylation causes a major conformational change, flipping the protein's orientation and releasing sodium to the outside. Next, potassium ions from the extracellular space bind to their sites on the enzyme. When the phosphate group is removed, the pump returns to its original shape, releasing potassium into the cytoplasm. Then another ATP binds, and the process starts again, up to 100 times per second in active cells. The right side of the image shows the atomic structure of the Na K ATPase. It consists of 3 subunits: the large F subunit that performs ATP hydrolysis and ion transport, the beta subunit, which stabilizes and positions the complex in the membrane, and a smaller Gang subunit that modulates activity. You can see the S subunit colored in cyan, winding through the lipid bilayer, with the beta subunit in gray, and the transmembrane helix of the Gang subunit highlighted in yellow. Also visible here is uabane, a plant-derived compound that binds tightly to the extracellular surface. Uabane and related cardiac glycosides, such as digoxin, inhibit the pump. By blocking sodium export, they increase intracellular calcium levels in heart muscle, strengthening contractions, an effect used in medicine for centuries. In summary, the Na K ATPase is a remarkable molecular engine. It consumes roughly 1/3 of the body's total ATP supply just to keep ions moving. Without it, electrical signaling, osmotic balance, and even cellular volume would fail.